Hi everyone, thank you so much for joining us this afternoon for the March of Dimes High School Science Week. We're so happy that you were able to be with us virtually today. I'm Monica Wirt-Parkinson, the Director of Public Programs for the Salk Institute. Today we're celebrating the 32nd annual March of Dimes High School Science Week. The program is a legacy of our founder, Jonas Salk. He believed in the importance of preparing the next generation to become great scientists and leaders. Dr. Christie Towers will be sharing about her career and work and answering your pre-submitted questions today. We hope that you will also join us for our other virtual programs this week, including a virtual laboratory demonstration featuring the Regulatory Biology Lab of Danielle Engel. Tune in to learn more about organoids and see how they are made. We also have a live career panel on Thursday, March 3rd at 4:30 p.m. Pacific Standard Time. You will have a chance to hear from 4 diverse Salk scientists and staff and participate in a live Q&A. In addition to our virtual programming for March of Dimes High School Science Week, we have an in-person paid internship opportunity for San Diego County high school students. Interns will have the opportunity to conduct real-life research alongside a Salk mentor. The application is currently open and closes on April 1st. For more information about all of our programs, as well as the internship program, you can visit our website at salk.edu/education or email our team at education@salk.edu. A huge thank you to all of our supporters of Education Outreach, especially the Ann and Neil Blue High School Science Fund, for making all of our programs possible. And now it is my pleasure to introduce Christina Towers. Dr. Towers joined the Salk Institute's NCI-designated Cancer Center in 2021 as an assistant professor. Christina's work has uncovered novel mechanisms that cancer cells can use to adapt to and circumvent the natural processes of the cell life cycle. She holds awards including the Cancer League of Colorado Pilot Award, the American Cancer Society Postdoctoral Fellowship, and many more. And now I turn it over to Dr. Towers to tell us more about her career journey and research.
I am so excited to be here today and have the chance to share with you guys a little bit about the work that I do here at the Salk Institute as a cancer biologist. But also, I really want to take the time here and get to chat a little bit about why I do what I do and sort of how I came to fall in love with science. So let's start there. I fell in love with science in the 2nd grade when I had to do this project where we had to draw a picture of every animal in the ocean. And that experience just made me fall in love with marine biology. And then all through grade school, into middle and high school, that sort of expanded into this love of all science, especially biology. So then when it came time to go to college, I decided to go to the University of Miami, and I majored in marine biology, and I was so excited to study whales, and I will never forget my very first intro to marine biology class, freshman year, and they take us out on the boat, and I learned very quickly that I get extremely seasick, and then had this very harsh reality that I probably could not do marine biology, and I changed my major within the first 3 weeks of college. And it was actually really sad because I love science, but I didn't know what to do with it. And so then I sort of explored what I think all of us that find ourselves in love with science do, which is becoming a physician. And so I spent a lot of time shadowing doctors, especially in the emergency room. And like I said, I went to college in Miami, so I was in inner city Miami hospitals. I would say almost all the patients that walked into these hospitals were either uninsured or underinsured. And I will never forget this woman that walked in with a breast tumor literally growing out of her chest, super late stage breast cancer. And it was the first time she was being seen in an emergency room because she didn't have insurance. That was just absolutely heartbreaking for me. I went home and cried myself to sleep. And again, realized that even though I love science, I didn't think being a physician, I just didn't have the heart for that. So again, I found myself at this moment of being like, oh, I love science, but what can I do with it? And I had an amazing freshman biology professor that was like, if you love science, you should try research. And I was like, what is that exactly, research? But he was awesome, and he got me into my very first lab. And so at I went into an immunology lab studying B cells, and I had no idea what research was, no idea what a B cell was, but I fell in love with it. I fell in love with this idea that every day we just get to ask questions, then design experiments to test those questions, but just generate 10 more questions. It was so fun, and from that moment, I was totally sold. I spent every second I wasn't in a classroom in the lab, to the point then when I went on to do summer research. And I took all of my summers, and I went to the University of Colorado, and I got to do research in diabetes and in atherosclerosis specifically. And so this is very different than immunology research, which is what I had first learned. And I think this opportunity to actually study totally different things, all still in the realm of biology, but this made me realize that my passion is not for something specific, but really just this understanding of how cells work and how it leads to disease. And how can we study that, and how can we come up with cures? So after college, after doing all this summer research, I then went on to get my PhD, and I decided to go to University of Colorado. I fell in love with it during my summers there. And so I went on to get my PhD, and this was my first time actually working in cancer research, and I specifically studied breast cancer at this time. And then after my PhD, I again stayed at the University of Colorado for my postdoc. So a postdoc's really similar to residency. So if you're familiar with when you're becoming a physician and you get your MD, you would go on to do this 5-year stint as a still part of your training and what's called residency. And so for PhDs, it's similar, but we call it a postdoc. So it's another 5 years or so where you've got your degree, you can do a lot of science on your own, but you still need that extra bit of training. And so for me, that's when I first started working on these recycling pathways in cancer cells. And still today, I work on those pathways, and now I'm here as an assistant professor at the Salk Institute and a principal investigator of my own lab. So I want to take a minute to step back here, and this is my path, this is my journey, and what I tried to add in were all these sort of curvy arrows, because when you think of it, it's just like, oh, she went to college, then she got her PhD, then she went to postdoc, but it really wasn't quite that linear, and there was lots of ups and downs, lots of wondering, is this really what I'm meant to do? Am I really cut out for this? Lots of conversations with different mentors along the way that supported me and encouraged me through this, and so during the Q&A at the end, we can talk more about that. But this is my journey, and here I am now as an assistant professor at the Salk Institute. And what I work on is a specific pathway called autophagy. I know autophagy is a really fun word, but it breaks down in the Greek to auto, which means self, and phagy, which means eat. So it literally means to eat yourself. So you can say it autophagy or autophagy, whatever you prefer. But what it is it's a cellular recycling process, and it degrades damaged material in the cell. And in the process, it recycles that damaged material into building blocks. So here's a little cartoon of the pathway. And so just to orient you here, we're looking at a cell. So this is the outer membrane of the cell, and all this green space is the cytoplasm, or the inside of the cell. So the way the pathway works is a vesicle is formed, and it engulfs or encapsulates all of this damaged material that's inside the cell. And then it creates this vesicle structure called an autophagosome, which can then fuse with another organelle in the cell called a lysosome. And when this happens, it allows all that material that's inside to be broken down and then recycled. And that recycled material fuels the rest of the cell growth and metabolism and survival. So one of the key genes or proteins that's important for this pathway is called the core autophagy protein, ATG7. So if we could delete or block this gene, we would be able to block the entire pathway. And if we block the entire pathway, then all of this material that should be getting degraded actually starts to accumulate within the cell. And this would be very bad for the cell, right? Because it's a lot of damaged material, it can create mutations, and it can make the cells very sick. So cells, especially cancer cells, will use this recycling pathway, autophagy, to help maintain their overall quality control, keep everything sort of status quo, right? So because cancer cells are especially dependent on this pathway, and to sort of just back up a little bit about cancer specifically, cancer, we think of it as like the superhuman cells, right? But really, cancer cells are actually in very harsh environments. There's not a lot of nutrients. They don't have a lot of access to oxygen or blood supply. And so being able to do pathways like this, where they can sort of break down or recycle their own material, it gives cancer cells an advantage to survive. So because of that, there have been a lot of clinical trials that are blocking this pathway with different drugs. I know this is a huge slide with way too many words to read, but the point is to highlight how many different clinical trials are now moving forward in all types of different tumors to block this pathway. And some of the results from these clinical trials have been really promising. So this is a graph that's called the Kaplan-Meier curve. And here we're looking at patients that have glioblastoma. And every single notch down here in this curve is when a patient dies from their disease. So if you look at the light gray curve here, this is the group of patients that were treated just with the general chemotherapy. But then if you look at the dark curve, this group of patients were treated with a drug that blocks autophagy. And you can see that this group, there are less notches down, right? So these patients are surviving longer when we treat with this sort of therapy. So this is fantastic news. It means that we might actually have a treatment that's working for these patients. But we're also seeing a lot of patients that are not responding, right? Still lots of notches down on this curve, patients that are dying from their disease. And so this really throws up a red flag, right? Which is, can cancer cells actually adapt when we block this pathway? And are there mechanisms of resistance to these drugs? And that's really what I study. So what are some of the technologies or techniques that we use in the lab to study this? Well, we do a lot of gene editing with CRISPR-Cas9 technology. And so how this works is you can think of it as a cut and paste for your genes. And so we can take the DNA and literally cut it and delete out the genes that we're interested in. And we use RNA molecules to sort of guide where these cuts will happen. So I take this sort of technology and I actually couple it with microscopy or imaging to watch what happens when we delete genes from cells. So I'm going to play a couple of movies here for you. So these movies are cancer cells that have two different colors in them, and we use these genetically labeled fluorescent proteins to give the cells color. So here in this first cell line, this is a cancer cell line, and it has a red color and a green color. And the red cells have a gene deleted that's important for autophagy, and the yellow cells don't. So if we play the movie, what you'll see is that over time, these red cells that don't have the autophagy gene, you can see they still grow. So you see lots of these red cells that have grown out. But now if we take a different cell line and we, again, with CRISPR technology, delete the same exact autophagy gene, now what you'll see is that only the yellow cells can grow. The red cells that don't have this autophagy gene, they can't grow. And so we can use this sort of technology to look at these different cells and say, okay, this type of cancer needs this autophagy pathway, but this type doesn't. And now we can study them and ask what's different. Can we figure out these resistance mechanisms? Okay, so now that I've given you a little bit of technology that we use, let's back up a little bit and ask, how can we use this technology to study a specific question? And that question is resistance to drugs. So if we zoom in on our tumor in a patient, we can see the tumor is made up of a whole bunch of different cancer cells, and we can zoom in even further to a single cancer cell. And within this cancer cell, we have all sorts of metabolic processes or pathways that are all happening at the same time. And the point of this metabolism is really to move molecules from one pathway to another, to create molecules or to break down molecules. All of that is encapsulated in the process of metabolism. So we could think of metabolism just like the highways or the roadways that make up our cities, right? Going all different directions, connecting with each other, moving things, creating things, breaking things down. So let's think of these roadways or these metabolic pathways in the context of one cell, but that exists within a whole tumor. So we could think of autophagy as just one of these metabolic roadways that we have here. So if we were to block the pathway, what would happen? Well, let's think of it as if there was a horrible accident on one of the interstates. That would create a huge traffic jam, right? All the highways are going to be blocked up because of that. So similarly, if we could block this pathway of autophagy, we can gum up the whole system. If we close down that road, the cancer cells are going to die because they need that pathway. And so you can sort of see that here by all of these grayed out cancer cells. But what we've shown is that cancer cells are really cunning. So think about when you're in a traffic jam, what are you going to do? You're going to pull your phone out and you're going to ask Google to reroute you, right? To find another way to get to work or to get to school. Well, we've discovered that cancer cells can do that as well. And they can actually create a detour almost and reroute their metabolism around the block. And this reinstates the whole metabolic network. And now the cancer cells can grow back and survive. You can see our tumor comes back to life. So what does this mean for our patients? It means that our patients that we thought were responding, we send them home, but then our patients are back in our clinics because their tumors have relapsed and they've become resistant to the drug therapies we were treating them with. So now what we're really trying to do is instead of blocking one pathway, can we actually identify all the pathways that the cells use, block them all at once, and once and for all cause the cancer cells to die to the point that they cannot come back? Okay, so how do we actually do that in the lab? So let's dive a little bit deeper into the data. So here we're looking at the growth data for the cells, for our cancer cells. So these curves are the growth curves as the cells grow over time. So here we're using CRISPR technology to knock out a gene that we know is not important for the growth of the cells. And that would correspond to this line right here. So the cells are still growing. But then we compare that to when we knock out a gene that we know is important for the growth of the cells. And that would be this line down here. And then what we ask is, well, how important is an autophagy gene like ATG7, which remember I showed you is important for the whole pathway to happen. So if we knock out autophagy, what happens? Does it look like the first line where they can keep growing, or does it look like the second line where they can't grow at all? And what we can see is that when we knock out this autophagy gene, it looks exactly like when we block a gene we know is important for growth. So this says that the cells are dependent on this autophagy process to survive. And when we block it, all the cells die. So this would correspond to the part in the diagram I showed you, right, where we put up the blockade, all the cells die, the highways are backed up. But then what we did in the lab was something really interesting. And even though all the cells looked really dead when we blocked this pathway, we left the cells and we kept changing, kept replacing their nutrients, giving them all that they could need. And eventually the cells started to grow again. And eventually we had enough cells that we could actually analyze them and look at how much protein do they have. And so this is what we call a Western blot, where we can assess how much protein is made from the genes. And so if we look at the ATG7 gene in the wild type cells, there's lots of ATG7, which is this black mark here. But our cells that we knocked out ATG7, you can see there's no ATG7 protein there. And they don't have any autophagy, which we use these readouts here, this LC32 molecule, as a sort of label for those autophagosomes. So they're not creating any autophagosomes. So then we can ask, well, if these cells that were really dependent on autophagy to grow, but now they don't have autophagy, how are they doing that? How are they surviving? So now we look at these cells, again, looking at the growth curves, and we can see that the cells that don't have any ATG7, they can grow exactly like the wild type cells that have all the ATG7 in the world. So this correlates to this part of the diagram here, right, where the cells now have detoured around and they can grow just fine. So of course, then the million-dollar question is, how are they doing this? So the how question is a really big question. So we wanted to break it down a little bit. So let's go back to what we know autophagy is important for. And autophagy is important for breaking down all of these different molecules. So instead of asking, well, how is it doing all of this? we wanted to sort of zoom in on a specific question, which is how is it breaking down mitochondria? So mitochondria is this little cartoon here, and it's another organelle in the cell, and it's a fascinating organelle because it's really important for creating all of the energy that the cell needs. And autophagy is important for breaking this down. So the selective degradation or the selective breakdown of mitochondria is not called autophagy, but instead we swap out auto for mito. You get it, mitochondria, mitophagy. It's a fun play on words. And we call this mitophagy. So our cells have been able to get around needing mitophagy, and they're growing, they've detoured, right? They're growing just fine. But are they really? So let's ask that question. So now this is a graph where we put our cells in a special sugar called galactose, which forces the cells to use their mitochondria especially. So it's a condition that really makes cells need those mitochondria to create energy. And in this context, our cells that don't have any ATG7, can't make any autophagosomes, they can still grow exactly like the normal cells. So this highlights, again, that these cells really have created a detour. They've gotten around needing the normal pathway, and they're doing it just fine, right? They're growing just fine, just like the normal cells. So again, how are they doing this? So what we discovered is that they are using these really fascinating structures called mitochondrial-derived vesicles. So looking at our cartoon of a mitochondria, so normally a whole mitochondria would get inside of an autophagosome. But what we've shown is that instead, these cells have been able to find a way to grab little bits and pieces off of their mitochondria, these little vesicles that sort of come off the mitochondria. And those can get degraded all by themselves without needing an autophagosome structure anymore. And we can image these with super resolution microscopy. So this is called electron microscopy that allows us to really look at the subcellular organelles in the cell. So just to get an idea of how small this is, this long line right here, this black line, that is 600 nanometers long. So 1 nanometer is 1/1,000,000th of a meter, right? B with a, billion with a B, right? It's a really, really small measurement. And so we're looking at a mitochondria with just amazing microscopy tools to be able to see this deep into a cell. And we can see these little vesicles that come off of our mitochondria. We can see our nice mitochondria here. You can see the inside structures that kind of look like what we see in our cartoon, and we can see these vesicles. So we can also use another type of microscopy called fluorescent-based imaging, where we can label different proteins with different colors. So here we can label the mitochondria with green and red to label these different proteins, and we look at them together. All of our mitochondria look yellow, and we can sort of zoom in this little box here to look at this imaging, and then we can draw what we call a region of interest and then quantify the signal of these different fluorescent markers to look at these different vesicles shedding off of the mitochondria. And so you can see here a structure that's just green or a structure that's just red. And we can count that signal of just green signal or just red signal. And this allows us to actually count these mitochondrial-derived vesicles and measure them and track them and see where they're going throughout the cell. Again, with the idea of being able, now that we know this is the detour pathway, this is what cells are doing, now can we go back with our CRISPR technology? Can we knock out these genes? And can we, once and for all, prevent the cells from growing? So sort of going back to the end all, right? What are we really trying to do? Is we're trying to identify what are these different pathways that the cells can detour around when we block autophagy. And we've identified that these mitochondrial-derived vesicles are just one of these pathways. But if we can really understand what they all are, we can design different drugs that target all these pathways, once and for all, causing the cancer cells to die. And this is how we will actually make a difference to our patients, right? This is how we will create drugs that actually block their tumors from ever coming back, saving lives and creating the cures. So with that, I will stop and just say thank you to my fantastic team here at the Salk. Also our funding that helps pay for this amazing work. I also have all of my contact information here, my Twitter account, my email, my lab website, so please check us out. And if you ever have any questions, you want to know what it's like to be a scientist, you just want to chat more about autophagy, whatever it may be, please reach out. We always love to talk science.
Thank you so much, Christy, for telling us a little bit more about your journey to Salk and your research. We have a couple of great questions that have been submitted from our students. The first one is from Granite Hills High School, and I think this is actually really interesting. What exactly is a principal investigator?
It's a great question. A principal investigator, or what we call for short a PI, is the person that is the head of the lab, that runs the lab and usually plays a big role in trying to understand or trying to figure out what are the big questions that we want to pursue. They're the ones that have the most knowledge of what's already been done and the most understanding of what needs to be done, right? So they're the ones that usually come up with the big questions, and then there are lots of students and postdocs and even physicians sometimes that work under them to actually do a lot of that research. But then PIs, especially at Salk and in San Diego, we really collaborate with other PIs from different institutions, but around the institute as well, to create sort of groups of labs that work together to really address big problems like metabolism and cancer. Not one group can do that, and it takes multiple PIs to sort of form that collaborative work together.
Great, thank you. Our next question is from a student at San Diego High School, and they're wondering, how do you maintain your work-life balance?
This is a great question. It's one that I would say I'm in the progress, of figuring it all out, as I think we all are. But for me, I have to have a balance. I have 2 small kiddos at home. And as much as I love science, it's not my everything. And so I actually try to create really strict boundaries for myself. I refuse to miss dinner. So, there are experiments that take long times, but I plan around my life at home as well, because that's really important. That's really important to me. And so, science does take a lot of thought, a lot of passion. But I think with any career choice, it's really important to make time for other things, because sometimes experiments don't work. Sometimes the hypothesis was wrong. And that can be really stressful. But having something to balance it out, I think really helps maintain your sanity for the long run. So for me, I work specific hours, and I really don't try to mix and match, although the pandemic's made that a little bit trickier. And then, sometimes when my kids go to bed or in between like basketball games on the weekend, I'll pull out time to make it into the lab or write grants and things like that. But really trying to keep my family and my science a little bit separate is what has worked for me.
Thank you for sharing that with us. Our next question is from a student at Hilltop High School. And you talked a little bit about CRISPR during your presentation, but they're wondering, to what extent are CRISPR and gene editing tools being used in cancer immunotherapy clinical trials? And are there certain cancers that are more responsive to gene editing?
Yeah, so the gene editing question, it's one that's on a lot of people's minds. CRISPR-Cas9 technology or genome editing technology really is pretty new. If you follow the news, it won the Nobel Prize just a couple of years ago, or last year actually. So it's really a new technology, and it's fantastic. There's a lot that we can do with it. There's a lot that we can do with it in the lab, like I showed you that we're doing, but there's also a clinical use to it. And what the student was alluding to was using genome editing itself actually in trials to affect the growth of tumors. And one of the ways it has been used for has to actually, in certain types of blood cancer, right, where we can actually do a blood transfusion, sort of pull the blood out of the patient, and then genome edit or knock out whatever those genes are in the specific blood cancer cells, get rid of that gene that's causing the cancer, and then put the blood back into the patient, right? So those are things that are being tried. I think there's still a lot to understand how that's going to work. Lots of off-target, things that we don't mean to edit. So we have to know what that is going to be. But I think really the larger question is genome editing largely, right? It can be a little bit of a slippery slope, and there's definitely some ethical questions that can arise with, if we can edit genes, well, how much should we be editing genes, right? Should we edit out genes that affect, you could think of things that affect like your susceptibility to specific diseases, right? So, we have patients that we know for breast cancer, for example, have a gene deleted when they're born, and this makes them more susceptible to getting breast cancer in their life. So could we say go into them as infants and change that gene? Well, genome editing technology says we could maybe do that, but should we do that? Becomes a little bit tricky, right? Because then it's like, well, actually, I just want to be taller. Can you edit my genes to be taller? I don't know that we should be doing all of that yet, but I think this technology is super powerful. It can definitely help us in the lab. I think it can help us in treating cancer and treating other diseases, but there needs to be a lot of understanding of how it works and regulations around it. And I think the whole scientific community is having those questions right now.
Great, thank you so much for that very complete answer. We appreciate it. We got a couple of variations on this question that students asked. And it's, if you were to give advice to your high school self, what advice would be? Any resources or programs that they should be looking into at this time to prepare them for a career in STEM?
Yeah, okay. Advice to myself. Don't worry about your hair so much. But no, true advice, I would say, when I was a high school student, I remember being unbelievably stressed about, looking back, what I would say were kind of small things, but in the moment felt really big. So I think my biggest advice would be, that one class that you're really struggling with, or that one AP test or exam that you have to take and you're super stressed about it, is maybe don't be so stressed about those individual things, because in the long run, what matters are the much bigger programs and experiences that you can be a part of. And so a single test, a single class, that's not going to make or break your career. But I would really look for opportunities that are immersive, right? Summer programs that you can be in the lab, year-long programs that you can interact even virtually with scientists, be able to shadow scientists, things like that. Those are the things that I wish I had access to as a high school student. And I really didn't. As I sort of shared in my journey, I really had no idea what research was until I got to college. And so some of these summer programs that we have here are just phenomenal. And I would really, really encourage you all to try to get into a summer program. And I know we have one here at Salk that is just absolutely phenomenal. And I know the applications have already launched and are live, and I think we talked about it earlier today. But please reach out to our program, but many others here in San Diego. And those sorts of opportunities, it's not just getting to do the science, but it's meeting the scientists. It's meeting the people that do the science, understanding what does it really mean, building a network of mentors that can help you along the way. I think that is so important. And just, that one test you're really stressed about, it doesn't matter.
Thank you for sharing that. This was actually a question that I was thinking of when you were giving your presentation, so I'm so glad that we had a student from Canyon Crest Academy ask it. So you talked a lot about autophagy and what happens in cancer cells. What happens in normal cells when autophagy is inhibited?
Yeah, this is kind of one of those million-dollar questions, actually, right? It's something that we have to think about as cancer biologists. Is you might have a tumor, say in your breast, but you have a whole lot of normal cells in the rest of your body, right? So one of the horrible parts about chemotherapy is that, yes, it kills the cancer, but it also makes our patients super, super sick. So when we think about these targeted therapies where we're trying to target a specific pathway, for example, autophagy, we really do have to think about what is it doing to the rest of the cells in the body. And so with autophagy is actually quite interesting because autophagy plays this role in quality control, right? Not allowing things that are damaging to build up in the cell. It can actually be a very healthy process in normal cells. And so blocking this healthy process is something we wouldn't want to do in a healthy cell. So part of what we are trying to understand is how do we know which tumors have this, I would say, addiction to autophagy, so that a low level of a drug, lower amounts of it would really hurt the tumor, but that amount of drug wouldn't be enough to hurt the normal cells, right? So we really are looking for situations, tumor types, where autophagy is going above and beyond in the cancer cells than it is in the normal cells, to try to create what we call a therapeutic window, to be able to block that autophagy without having so much of an effect on the normal cells, because we do know that it is important in the normal cells. So it sort of adds to the complicated problem here, but it's something that we're all working on.
Great, thank you. And we had a student from Long Beach High School ask a question, which I think a lot of students think about when they're thinking about a career in STEM, is what does a day in your life look like when you're working in the lab?
Yeah. Well, I will say one of the great parts is that every day is different, which makes it really fun. But I would say a normal day is coming into the lab. It's checking all of my cells that are growing. I grow a lot of cells in an incubator, I check them to make sure that they're growing and healthy, and these are cancer cells. I might set up an experiment where I would treat those cells with different drugs. I might take them down to the microscope and image them. I might try to put different drugs to see how much death I can see through this imaging. And then I would collect those images or that data. I would take it back to my computer, sort of pull up the images, look at them, do a lot of calculating and quantifying of the data. And then I would interact with my students that are in the lab to try to look at the data all together and say, well, what do you think this means? And they would, it'd be a back and forth with them saying, oh, did you read this paper that just came out that showed this? Maybe this is what's happening. Oh, maybe we should go talk to this PI down the hall. I bet they can test this question. So there's a lot of brainstorming back and forth. And then I'd say at the end of my day, there's always some time that goes into writing grants, trying to appeal to different groups that might want to fund this research, because the truth is we can't do any of it without funding. So there's a lot of training, there's a lot of experimentation, there's a lot of writing and thinking, hypothesizing, which keeps it really fun.
Great, thank you for sharing that. Our next question is from a student at Granite Hills High School, and they're wondering, how are new cancer treatments considered? For example, are the new therapies built off of improvement and refining existing ones, or do researchers just kind of throw everything in the kitchen sink at it to see what works?
I know sometimes it feels really overwhelming, or maybe even sad that, we're so much technology and we still don't really have a cure for cancer. I know that can feel really depressing to everybody that has probably had a loved one suffer with this horrible disease, but it's also really sad to us as the researchers, right? We've been working at this problem for so long. But what I will say is that techniques and technologies really are pushing forward. And the way that we decide how do we take a therapy that we think will work, how do we move that into patients? Well, the truth is, any drug that's in a patient, the pipeline right now, it's about 10 to 15 years. So any drug that's in a patient, 15 years ago, someone discovered that drug targets a specific pathway. It blocks autophagy, for example. And 15 years ago, they figured out how it works in cells in the lab, and they figured out, well, if we treat it this way, or if we add this dose. And then they took those and they usually ran preclinical trials, oftentimes in models in the lab. So sometimes those could be animal models, sometimes they can be cell culture models, to really understand the dosing. And then that work is published. And when work gets published in the scientific community, it allows hundreds of thousands of eyes on that work to say, actually, that makes no sense. What were you doing? Or to say, we saw similar things in our lab and we showed this the same way. And so the work then gets corroborated by a lot of people around the world. And so by the time something goes into a patient, it hasn't been shown one time. It hasn't been shown by one group. It's been shown by many, many different groups around the world that have all validated that work. And that's why what we call peer review is such a critical part of the scientific community, because we need others' input. People come from different points of view, from different backgrounds, and we really need all of that information to say, this is what's really going to work. And the truth is, even then, they don't all work. And, the reason for that is because cancer is a very complicated disease. And it's not like, for example, COVID-19, which is one virus that's the same in every patient. In cancer, every single person's cancer is different because every single person is different. So it's a lot harder to target it because we have to come up with sort of a different therapy for each person. We're trying to figure out how can we group people and things like that, but it's really a complicated disease. But I am 100% confident that we have the techniques, the technology, and the brain power and the willpower to beat this thing.
Thanks so much. Our next question is from a student at High Tech High, and they were wondering, what interventions are you testing that prevent cellular recycling, and how do these exactly work?
Yeah. So I alluded to at the beginning of my talk some of these clinical trials that are blocking these autophagy pathways in people, in patients, and the drugs that we use are specifically lysosome inhibitors. So remember in the pathway, the autophagosome that has all that damaged material, it goes to the lysosome. And the lysosome is what actually breaks all that down. So we use drugs now that specifically target the lysosome so that the material can't be broken down. So there are a lot of different drugs that are starting to move into clinical trials, but we're also really trying to come up with better drugs because the lysosome actually does a lot of things. So targeting the lysosome is not specifically targeting autophagy. And so there's more work now being done. Can we target ATG7, for example? Can we come up with drugs that would really hit that gene specifically, or even other specific autophagy genes? So there's a lot of work to try to diversify the types of drugs that we're using that we think may work better than just broadly targeting the lysosome.
Great, thank you. And we have one last question from a student at Mark Twain Mesa. And they were wondering, you touched on this a little bit when you were talking about your journey, but is there someone who inspired you to become a research scientist, a mentor?
Yeah. For me, it's not just one person. It is a handful of people. Probably starting with my grandfather, who was a chemist, and I just can remember him standing in the back of the room whenever I gave a presentation on anything, just like coaching me through it. So definitely I do a lot of it for him. And he's the one that really fueled the flame of my, fueled the flame of my passion when I was growing up in science. But then, to be honest, he didn't really know the opportunities that were out there. He couldn't really mentor me in terms of actually getting into STEM opportunities. And so it was really my professor in college that first gave me opportunity in the lab. He was one of the big ones. And then it's really been key mentors all along the way of my journey. And one of the biggest pieces of advice I would give anybody at any stage, whether you are the second grade me, or you are the high school me, or you are, the PhD me, I would give the same advice, is that you have to find people that advocate for you, people that will mentor you, that will help you find the path, because to be honest, it's not a straight path and it can be tricky. And so finding people that will help you along your journey, there really is no value to it. There's no value to that. So getting into programs, getting into summer programs where you get access to scientists and mentors that can help you along the way, I think that really is the key to being successful in any STEM field.
Great. Thank you so much, Christie. And thank you so much, all of you, for joining us today. We were so glad to learn a little bit more about your journey to Salk and your research. And just a reminder, you can visit our website to both view our education outreach programs as well as to take a look at Christie's lab to see what else she's working on. So thanks so much, and we look forward to seeing you at our upcoming programs the rest of this week.